Good morning, everyone. Welcome to our plenary session, Moving PF Care Forward, Updates from Ongoing Research. This is a critically important topic for patients and families, and we are very fortunate to have a highly accomplished and influential national health research leader with us today. He has also been a very busy person during the pandemic, for which we owe him and his team a debt of gratitude. Dr. Gary H. Gibbons is director of the National Heart, Lung, and Blood Institute, NHLBI, at the National Institutes of Health, where he oversees the third largest institute with an annual budget of approximately $3 billion. The NHLBI is the largest public funder of pulmonary fibrosis research in the world. The Institute provides global leadership for research, training, and education programs to promote the prevention and treatment of heart, lung, and blood diseases. Since being named director, Dr. Gibbons has enhanced the Institute's investment in fundamental discovery science, steadily increasing the number of awards for both established and early stage investigators. He has demonstrated his commitment to nurturing the next generation of scientists by expanding funding for career development and loan repayment awards, as well as initiatives to facilitate the transition to independent research awards. Dr. Gibbons earned his undergraduate degree from Princeton University and graduated magna cum laude from Harvard Medical School. He completed his residency and cardiology fellowship at the Harvard-affiliated Brigham and Women's Hospital in Boston. He has been a member of the faculty at Stanford University and Harvard Medical School. At the Morehouse School of Medicine, he served as the founding director of the Cardiovascular Research Institute, chairperson of the Department of Physiology, and professor of physiology and medicine. Dr. Gibbons also served as a member of the National Heart, Lung, and Blood Advisory Council before becoming director. Dr. Gibbons has received numerous honors, including election to the Institute of Medicine of the National Academies of Sciences, selection as a Robert Wood Johnson Foundation Minority Faculty Development Awardee, selection as a Pew Foundation Biomedical Scholar, and recognition as an established investigator of the American Heart Association. On a personal note, I had the opportunity to meet Dr. Gibbons at a PF patient education event hosted by the University of Minnesota a couple of years ago, and came away very encouraged by his obvious personal commitment to moving the PF research field forward as rapidly as possible. Again, we are very fortunate to have him join us today. During this morning's session, we will also be joined by our esteemed panelists, Dr. Fernando Martinez from Weill Cornell Medicine, Dr. Elizabeth Freiheit from the University of Michigan, Dr. Ayodeji Adegunsoye from University of Chicago Medicine, and Sean Keoni Craig, a lung transplant recipient who appears in the PFF's new lung transplant journey video. So without further ado, please welcome Dr. Gary Gibbons.
Greetings. On behalf of the National Heart, Lung, and Blood Institute, it's our pleasure to be joining you as part of the Pulmonary Fibrosis Foundation during this summit, where we can share the NHLBI's portfolio relevant to addressing idiopathic pulmonary fibrosis. This audience is very familiar with the challenges posed by the diagnosis of idiopathic pulmonary fibrosis. We recognize that this is a complex disorder with a very high 5-year mortality rate, a variable clinical presentation, and clinical course. We recognize also that there's a need for accelerating new therapies that we hope eventually can lead to the prevention and preemption of this disease. Toward that end, it's critical that as we make advances, that we promote scientific discovery that is part of this virtuous cycle of innovation to improve outcomes for those living with pulmonary fibrosis. This virtuous cycle depends on basic discovery science that understands the pathobiology and molecular mediators of pulmonary fibrosis, and then paves the way for the identification of new targets for therapeutic interventions by having that molecular understanding of the disease. These then can be tested with novel therapeutics in early stage and late stage clinical trials to see what's safe and effective at changing the clinical course of pulmonary fibrosis. It builds on this notion of having targets and animal models that give us insights, as well as studies in humans that define the molecular mechanisms of disease progression. And ultimately, as those are tested, to have them implemented in clinical practice in ways that changes patient care and delivers better health outcomes for those with pulmonary fibrosis. Toward that end, we know that there are patients perhaps already participating in the summit who are struggling with the latter stages of the disease. And we also recognize that we want to do things that will improve the quality of life of those living with IPF, as well as a pathway toward developing new therapeutics and ultimately precision medicine, where we can more effectively prognosticate who's at risk, who's most likely to progress, and to provide the right drug at the right time for the right patient to again predict, preempt this disease ultimately. We're encouraged that we are making progress. One of the things that's clear is that delayed diagnosis and delayed treatment is one of the challenges facing patients with IPF and the clinicians who care for them. When we recognize that those delays can reduce the window of opportunity to make interventions that can change the natural history of the disease. And so there are promising new imaging technologies that we believe may provide an earlier insight and diagnosis. For example, in the NHLBI's intramural program, there have been some exciting new advances in magnetic resonance imaging technology, as shown on the right side there. These new techniques are providing greater resolution of lung pathology that may enable us to diagnose conditions like IPF much earlier. And that gives us hope for earlier diagnosis and potentially earlier interventions to change the clinical course and progression of this disease. Similarly, many of you may already have experienced, those patients in this audience, some of the benefits of pulmonary rehabilitation that is having a beneficial effect on the quality of life. And we look forward to expanding upon the results as shown on the left that indicate a short-term benefit, and looking forward to extending these observations to see what interventions can further sustain those benefits over the long term, as well as understand how we can best ensure that these treatment benefits reach all patients who need them, no matter where they live, and using different modalities. And so this is an area of ongoing research. Toward that end, we also recognize how many patients struggle with the supplemental oxygen, the lifeline. And this poignant quote from an IPF patient, needing oxygen is like being in jail. And I know from personal experience, not only as a clinician, but my brother-in-law, who also is on continuous oxygen, gives me that patient perspective of what a challenge this represents, and how little innovations that's occurred over decades in this space. And that's why the Institute is launching a challenge prize, if you will, to incentivize academics and researchers and entrepreneurs to consider ways to innovate, to enhance the livability and development of new techniques for supplemental oxygen that are more portable and more user-friendly. And so those technologies are something that, again, we want to catalyze as part of the Institute efforts to enhance the quality of life of those living with pulmonary fibrosis. Let me shift now to accelerating our scientific priorities in pulmonary fibrosis with a particular emphasis on discovering novel therapeutic. This begins as part of that virtuous cycle with discovery science to really try to elucidate what are the molecular basis and drivers of pulmonary fibrosis that leads to that seemingly progressive decline in lung function. And progress is being made. Indeed, some of the mediators of that process are being revealed. And that gives us promise for the development of targeted therapeutics to interrupt the pathobiology of the disease in ways that we hope can preserve lung function and again, prevent the inexorable decline. Toward that end, they're revealing clues that inflammatory cells participate and play an important role, that the regenerative capacity of the lung to respond to injury in some patients may be compromised, and that may predispose to increasing fibrosis. And of particular note, we're starting to elucidate with NHLBI funded investigators the drivers of the fibrosis, the scarring that's occurring in the lung that compromises lung function, makes it so hard for patients to function and breathe effectively. And we believe this is a promising area for new therapeutics, particularly related to transforming growth factor beta, TGF-beta, as a key driver of that fibrotic process. Again, basic science and discovery science is providing new and exciting clues along this line of defining the molecular mediators of IPF. In particular, a cousin, if you will, of transforming growth factor beta, connective tissue growth factor, does what almost its name implies. The fibrosis, the connective tissue accumulation that scars the lung is driven in part by factors like this generated in the lung. And indeed, in studies in mice where we can manipulate these mediators, you can see that there's increased expression of CTGF and it induces fibrosis in a way that mimics what we see in patients with both inflammation and accumulation of fibrous tissue and collagen. And indeed, what we're seeing in the mice appears to also be happening in humans, where patients with IPF are also showing some signs of activation of this pathway. So again, this provides us with further confidence that we're starting to hone in on some of the key mediators of this disease. Similarly, as we develop these targets through these studies of discovery science and pathobiology, both in animal models and in human tissues and studies, this provides the preclinical basis for advancing drug development that can target these mediators and develop drugs that can actually interrupt these pathways. And indeed, some of that basic science research investment appears to be paying off as part of the public-private partnership of that virtuous cycle where the private sector and pharmaceutical companies are now developing agents that target that same molecular axis of TGF-beta, CTGF, and indeed this agent shown here is showing some early efficacy, appears, in preventing some of the decline in pulmonary function in patients with pulmonary fibrosis by inhibiting CTGF. And so this is very exciting and promising. This is only phase 2 data, and the phase 3 efficacy trials are underway now. And so we believe that there are some wonderful options and promise of therapies on the horizon. Let me then close by pivoting to where we see the future continuing to unfold as we move toward precision medicine. This is where we hope to get a better understanding of IPF, understand its full clinical spectrum and clinical course, and recognize that there actually may be subsets of patients within this large category of pulmonary fibrosis, and elucidating that by leveraging, again, some of the technologies that exist, in particularly genomic technologies. They're able to help us understand different patient subgroups captured within this large diagnostic category of IPF. And this is part of personalized precision medicine, where we can get the right drug in the right patient subgroup at the right time window to actually hopefully reduce the progression of disease and ultimately hopefully preempt or reverse the disease. One of the areas of this new molecular technology is to be able to understand these molecular pathways even down to the single cell level within the lung over time in disease and in health. And the NHLBI is building such molecular atlases to tell us about the normal gene expression profile of all the cells in the lung under normal circumstances and how they're perturbed in various disease states of the lung, whether it's IPF or chronic obstructive pulmonary disease. And again, these are showing promise in the future. Indeed, some of those studies done in patients with IPF are already characterizing some of the pathways. Indeed, this study is provocative in indicating, again, the role of the TGF-beta as a key signaling pathway that appears to be modulated in this context. And in fact, as shown on the right, the pattern of gene expression at the single cell level may even be giving us prognostic information of who is most likely to progress as opposed to who is relatively stable, something that clinicians and patients often are unclear about as to what their prognosis can and could be. Moreover, this may start to identify distinct patient subsets that may respond to particular therapeutic targets and drugs. At a larger scale, this fits in with our TopMed program, Transomics for Precision Medicine, that the NHLBI has pioneered and established, one of the largest data sets of genomic information and clinical phenotypes that exists in the world, particularly one that has a diversity of ancestral groups within the human family. Indeed, this includes healthy individuals as well as diseases, both common and rare. And certainly a high priority for the NHLBI was to ensure that samples from patients with idiopathic pulmonary fibrosis were part of this data set. And indeed, we believe it will pay dividends in helping to elucidate this disease. Indeed, there seems to be progress already being made along this front. With these large data sets, we're able to get a sense of the genetic predisposition to certain diseases like IPF. And one of the early hits with these genetic association studies was for the MUC5B gene in association with pulmonary fibrosis. This provided some clues as to one of the molecular pathways that contributes to this disease and the genetic susceptibility that might explain some of the clustering in families, et cetera. Toward that end, again, as you can see, there's evidence of changes in MUC5B expression in patients with IPF that again gives us even more confidence this may be a target and a marker of disease and processes. Indeed, in going from the bench to the bedside and then back to the bench again, we're able to validate whether some of these targets picked up by genetic association are really driving the disease. And again, this is where going back to an animal model can help provide, again, further assurance and validation that we're on the right track. And in this regard, if you take that MUC5B gene and that variant, in fact, it can recapitulate, again, some of the components of IPF, even in a mouse. And you can see on the left image there, the normal, but also on the right image of the mucus buildup that's occurred in the mouse given that variant shown in that genetic association study, where there is that accumulation of mucus. And that's telling us that we're starting to piece together the factors and drivers of the various manifestations of IPF. Moreover, as shown on the right-hand panel, this also gives us a novel animal model related to IPF that then can be used to test various therapeutic interventions. And indeed, in this case, defining new mucolytics that can address that accumulation in a way that, again, could have therapeutic value. These are early days, but by having some animal models that really are getting closer at recapitulating IPF, that's what gives us confidence for future development of new therapies. This approach continues, I believe, to yield value. As shown on the left-hand panel, these studies have extended and expanded, particularly under TopMed, our program. And this new analysis not only confirms the previous one that MUC5B is an important potential mediator, but there were a number of other hits and discoveries as part of this, one of which is this gene called DEPTOR, which was picked up very recently as a new IPF risk DNA variant. And DEPTOR is notable because of its interaction with important pathway, mTOR, that's important in biology and metabolism, but also it's linked to that same mediator, TGF-beta, in which TGF-beta appears to modulate DEPTOR expression, and again is reinforcing the same pathway. And so again, when we see this convergence of results, all pointing to the same thing that appears to potentially play a key role in fibrosis, we get a sense that we're now identifying more and more mediators along particular pathways that might be amenable to therapeutic interventions that could potentially change the natural history of the disease. So I hope what we've tried to do is to convey some sense that the NHLBI is committed to understanding idiopathic pulmonary fibrosis as an important scientific priority, and that we have a broad portfolio of research that's attempting to enhance the quality of life of those living with the disease, as well as look to the future in developing a whole pipeline of new therapeutic targets and new therapeutic drugs aligned with those targets that we hope in the context of the future would be used to predict, preempt, reverse, and certainly arrest or slow the progression of this disease. That's our vision, and we believe we're making progress toward turning that into a reality. And with that, I'll end. Final slide, which is to recognize that the NHLBI fulfills its mission to turn discovery science into the health of the nation through a circle of partners. And certainly the Pulmonary Fibrosis Foundation is a key partner in that process. I remember quite fondly visiting the summit a few years ago, where Paul was kind enough to even pick me up from the airport. And he shared with me that he would donate his body, literally, toward the understanding of IPF. And I hope that in the talk today that we've tried to live up to the commitment we made to Paul, that we wanted to look forward to a day when we didn't call it idiopathic pulmonary fibrosis because we didn't know what was causing it. But we actually would find it out in a way that could change treatment. We hope and look forward to living up to that commitment every day. Thank you for your attention.
Hi, my name is Fernando J. Martinez. I'm the Chief of the Division of Pulmonary and Critical Care Medicine at Weill Cornell Medicine. I've been asked to give you a quick summary of the paradigm for the Precisions trial, and I'll try to do that in a relatively short period of time. There are a series of conflicts that I can mention here. None of them should really relate directly to this presentation. And the overall goals of the Precisions program is really to try to address the idea of precision or personalized medicine, which is really the standard in the oncology world in the area of ILD, and really translating this directly into an actionable approach that we can provide our patients and their providers. So I will briefly describe to you what Precisions is as an overall goal. And it's based on the concept that interstitial lung disorders are really a series of syndromes that share a lot of common signs and symptoms. And we want to be able to help guide our patients, their caregivers, with regards to how to be as accurate as possible in ensuring a correct diagnosis, try to ensure that we have a correct prognosis, and most importantly, to lead therapy in a precision or personalized fashion. To do that, a group of us approached the National Institutes of Health to allow us to address three specific aims in the entire program. The first aim is actually a therapeutic trial. It's the first of its kind, genotype-driven pharmacogenetic trial, and aims 2 and 3 really reflect molecular characterization of a broad range of patients with varying interstitial lung diseases. And I'll touch briefly on all three of those, but I'll spend most of my time on that aim 1. So to give you a graphical representation of what we're trying to do, aims 2 and 3 you see on top are aims that leverage the patient care registry of the Pulmonary Fibrosis Foundation and a series of global cohorts from our collaborators to perform a very detailed transomics approach, including proteomics, transcriptomics, we're actually including metabolomics, genomics, and that'll be paired with whole genome sequencing in a population of patients. The goal is to be able to provide all of us a sense of how to molecularly characterize patients and expand ourselves just from a nosological approach of saying you have disease A versus B, but exactly the molecular characteristics that define an individual patient with an ILD. And then aim 1 is this pharmacogenetic trial that focuses on one of the ILDs, idiopathic pulmonary fibrosis, because it's where we have the best data. So let me spend a little bit of time talking to you about this pharmacogenetic trial. The basis of this goes back to the Idiopathic Pulmonary Fibrosis Clinical Research Network, which was an NHLBI sponsored trial, series of trials about a decade ago. And one of those trials tested a very cheap intervention, N-acetylcysteine or NAC, in idiopathic pulmonary fibrosis. That was based on extensive prior data, a lot of it from our European colleagues. And in this particular study, where NAC was compared to placebo, there was no change, as you can see in this slide, in the loss of lung function, a typical measure of progression for our IPF patients. So we were disappointed when we saw the overall results. However, the editor of the New England Journal of Medicine had asked us to take this, what appears to be a very complex figure, and to present it in the main body of the manuscript. So the diamonds, these triangles are all of the patients that were enrolled, but this study was actually enrolled in 2 phases. And there was a phase when the trial, which was called Panther, had 3 arms, one of which was an arm of triple immunosuppressive therapy. A second arm was the NAC alone. And that original arm of immunosuppression was stopped early. And so about half the patients in just the NAC versus placebo comparison had been enrolled. And this is the primary endpoint, and then a whole series of secondary endpoints. And interestingly enough, most of the endpoints actually favored the NAC versus placebo. The study was modified when the triple therapy, the immunosuppressive arm, was removed, and we enrolled about the next 40% of the patients at the same sites, same inclusion criteria, same NAC from the same manufacturer. And in this time, almost all of the endpoints went in the other direction. And we were puzzled why there would be such a dramatic difference between patients enrolled with the same criteria, same diagnosis, same centers, same techniques, everything were the same between those patients originally enrolled and the patients that were subsequently enrolled. And there was no obvious characteristic that differentiated those patient groups. It ended up being that there was actually a genetic imbalance between the patients enrolled early in the trial and later in the trial, with particular genotype, this TT genotype of TOLP, which is a toll-like receptor signaling pathway, which actually has a strong mechanistic interaction with NAC. And if you had this particular genotype, the NAC patients seemed to do quite a bit better than if you had been on placebo. If you had this genotype, it was reversed. It turned out that this genotype was preferentially enrolled early in the trial. This genotype was enrolled preferentially later in the trial. And so we were able to confirm this interaction with TOLP in a subsequent series of cohorts. That led us to the thought, you know what, we think the reason this study ended up being negative was actually a very important mechanism. It was that the patient's own genetic structure interacted with NAC in regards to the clinical outcome. This would be the first time we would have a strong genetic pharmacotherapy interaction leading to a treatment result. But that could not be proven in this analysis. It could only be proven in this kind of a trial, which is a trial where we preferentially enroll the patients that have this particular gene structure for that particular genotype, where we decide, does NAC work versus placebo? So we're essentially looking at this population of patients. So this is a pharmacogenetic trial. To be able to conduct this trial efficiently, we structured what looks like a very complex overall structure, but it actually has some several very important components that I think are also quite innovative. It has a committee of individuals that run the study. There's a data coordinating center at the University of Michigan, the SABRE group. And then there's a clinical coordinating center that's run by me and my co-PI, Imre Noth. And we've strongly leveraged the Pulmonary Fibrosis Foundation and its registry, all of those sites, to be able to define a study that we think, and I'll show you, is proven to be very efficient. We also had a strong partnership from an independent foundation, the Three Lakes Foundation, which has had not only financial contributions, but scientific contributions to the way this study is being designed. So this is what we ended up deciding to do with the partnership with the PFF. You're all quite familiar with the Care Center Network and its registry with large numbers of patients that have consented to have their samples collected and stored or analyzed. And so for AIMS 2 and 3, where we're looking at this molecular characterization, we're using data from many of the patients enrolled in the PFF sites. But for the therapeutic trial, we really are targeting a very specific population of patients. And a way of getting this done very efficiently was to say, you know, if we take a subset of these care center network sites, they already have recruited patients with IPF who have already agreed to provide us with DNA and genotyping, and we'll start enrolling those patients that are already in the registry. And then as the new patients come in, we will genotype them, screen them for that genotype, and enroll in that fashion. This ability to enroll a group of patients upfront from the beginning of the trial, and then as the trial went on, has proven to be a very efficient format. It all got waylaid by the pandemic. The neck preparation that we use comes from a pharmaceutical company that's based right up here around Milan, which is where we had the major initial European pandemic. So everything was delayed by months. Despite that, we've actually been able to catch up, and enrollment is actually proceeding very nicely in this very unusual trial. We've tracked off a little bit because when you actually look at the enrollment reports, this is the most recent one from 5 days ago, that overall enrollment is here. And you can see that all of the enrollment so far has come from just the original 7 sites. We've got 18 more sites that are going to be brought up now in the next few months. But just the first 7 sites have been able already to enroll 32 subjects. And it's very interesting that the idea of enrolling patients that were already in the registry with DNA available has been about half the subjects. The ability of enrolling patients that are being genotyped in real time as they present has been the other half. So that approach of how the study was structured in close partnership with the PFF, it seems to be working. And actually, the proportion of individuals that have that favorable genotype of 26% is exactly what the preliminary data from the IPF net suggested. So overall, this idea of structuring a comprehensive modality to molecularly characterize patients and to perform a genotype-driven pharmacotherapeutic trial in IPF, which with a structure that includes very closely PFF centers, is working and is working very efficiently despite the pandemic. So I will leave you with the following thoughts. It's my belief that the era of precision or personalized medicine in ILD is here. And as this study progresses and provides more information, it'll become much more codified. The Precisions program not only will characterize a broad range of patients for their molecular appearance, it will provide us with new therapeutic targets. The idea of conducting a therapeutic trial with all of these partners, in particular the PFF, has proven to be a very efficient new paradigm for very innovative studies of IPF. And I really do believe that this entire Precisions program and all of these partners are really establishing a very robust format that I think will dramatically alter the future of how we manage our patients from diagnosis to prognosis to therapy. Not only doing that, but also establishing a really unique platform by which therapeutic trials can be rapidly and efficiently conducted. I'm sorry we couldn't be there in person to do this, but thanks for listening.
Hello, my name is Elizabeth Freiheit. I am an associate director at the SABR Research Group at the University of Michigan, and I'm also the DCC director for the PFF Registry. The title of my talk, Research Results, might lead you to believe that I'll be providing more results, as Dr. Adegunsoye did in his portion of this plenary session. In 13 minutes, I can't cover all the exciting research going on in their various stages of development, from proposal to analysis to abstract to reanalysis and to publication. So my goal for this talk is to provide a sense of the maturity of the registry database and the types of research that we at the DCC are involved in collaboration with investigators around the country. SABR is a unit in the University of Michigan Department of Biostatistics. We serve as a data coordinating center for the PFF registry, as well as many other studies. We have a significant portfolio of pulmonary trials and studies in addition to working on the registry. As a data coordinating center for the PFF registry, we have a central role in its operations. We design the database and the data entry and data collection systems. We contract with the CCN sites and help with regulatory requirements. We assist coordinators in entering the data, answering questions about data collection. We also have coordinated the collection of the biosamples and the distribution to researchers of the biosamples. We've contracted with National Jewish Health and are coordinating now the distribution of high-resolution CT scans to researchers. We also collaborate with study investigators on statistical analyses, and we're involved in the design of the new community and CCN registries that we're working on rolling out at this time. Our involvement with the PFF registry began in 2015 when we assisted with the design of the registry. The first participant was enrolled in 2016. In 2017, there was enough data to begin a preliminary look at the data, so we initiated our first ancillary studies at that time. In June of 2018, we completed our enrollment, having consented over 2,000 patients. This past summer was an important benchmark for us because it meant that now we had at least 3 years of follow-up data on all active participants in the registry. At this point, the data has matured enough that we can really start looking at factors associated with long-term disease progression, change in quality of life over time, and survival. Since the first ancillary studies were initiated in 2017, SABR's been providing researchers with data sets and biosamples, and also with data analyses. Much of the research that was initiated a few years ago is still ongoing because we've been waiting for that longer-term data to be available in order to thoroughly analyze the various topics and hypotheses under examination. It's a long process. Typically, there's an initial analysis, which becomes a poster or presentation at a medical conference. Then the analysis expands in order to answer more questions on that topic. Sometimes we re-pull the data to get more updated information and to add more long-term data. This time, the reanalysis will be shared with the medical community by being submitted to a journal for publication. We are at this stage with many of our studies right now. One of our most precious resources are the biosamples donated by about 9 out of 10 participants in the registry. DNA, RNA, plasma, and serum for nearly 1,800 patients with pulmonary fibrosis is a precious commodity indeed. But what makes it almost priceless is the fact that these samples can be associated with over 3 years of clinical follow-up data. We have about 10 study investigators right now who all have received samples to examine a number of topics. This fantastic combination of the sample donation and the clinical data allow us to answer the following questions. Can we use protein markers or genotypes found in the biosamples to improve our ability to make earlier and or more confident diagnoses? As we know, a correct and early diagnosis is critical to applying appropriate treatments to stop progression and to stabilize or even cure the disease. Can we use biomarkers or genotypes to predict who is likely to progress quickly? This would help us determine how aggressively we need to be treating the disease for an individual person. And finally, can we identify treatments that are more likely to be successful? Precisions is the name of a randomized clinical trial to see if people with a certain genotype are more likely to respond well to a particular drug that is suspected to work well in that group. Other studies are also looking at the role of particular fatty acids, of certain proteins, and of certain gene expressions in disease progression and survival. Our newest richness in data comes from the collection of high-resolution CT scans by our partner, National Jewish Health. Our clinical sites have been uploading available CT scans to a central repository. These scans are scored and graded by central radiologists. These data are just now, this fall, available to all researchers who receive approval for their research proposal by the PFF Ancillary Study Committee. This research should also help us refine our diagnosis process, improve our ability to predict disease progression, and identify treatments that are more likely to be successful. We have a number of ancillary studies that have started and will use the HRCT scans. Some researchers at National Jewish Health, Drs. Humphrey and O, have just begun working to address these first 2 questions in particular. Dr. Smith and Kulkarni at the University of Alabama are developing a new predictor based on CT scans. Dr. Aaron and Dr. Gupta at Genentech will draw associations between biomarkers from CT scans and biomarkers from biosamples to use them to try to improve our ability to predict disease progression. Another great source of data richness are the 4 surveys completed by our participants every 6 months. These questionnaires give us a window into their quality of life and help us to flesh out their experiences beyond clinical data. In particular, experience with cough, shortness of breath, fatigue, and general quality of life. Many studies that have focused on other outcomes still find it informative to compare quality of life scores between the groups that are being compared. And these scores have been incorporated into a great number of the many ancillary studies that are mentioned in this presentation. One study in which the quality of life has been front and center has been conducted by Drs. Janet Lee of Utah and Nina Patel of Columbia. They have taken a deep dive into characteristics that predict severe cough and also have looked at the role of cough as a surrogate for disease severity. Other researchers, such as Dr. Sachin Gupta at Genentech, are now working with us to look at the impacts of treatments such as oxygen and antifibrotic use on quality of life over time. Lung function and survival are the most important data we collect. Nearly every analysis we do investigates factors that are associated with maintaining lung function and living longer. We get this information from site coordinators who enter information from medical records into our database every 6 months. Information such as lung function tests, lung performance tests, hospitalizations, exacerbations, and other updates to health status provide this important information. The typical research question associated with lung function survival is this: what factors are associated with the loss of lung function and long-term survival? Some examples of research that have been done are the ones listed here. Dr. Podolanchuk was looking at proteins found in the blood to see what might be affecting outcomes, certain proteins. Dr. Lee was looking at people who maintain body weight versus people who lose or gain body weight and their association with function and survival. Dr. King was looking at anticoagulant use and its effect on survival. And Dr. Berg was looking at oxygen use and its effect on survival and hospitalization. In addition, there are other topics being studied now, such as the effect of air pollution and biomarkers, the effect of hospital stays, the effect differences between rural and urban dwelling patients, and then racial disparities. You saw the results from Dr. Adegunsoye's presentation as well. We're also looking at differences in practices around treatment. How do people taking antifibrotics differ from people who are not taking them? Dr. Holtz at University of Michigan found regional differences in what was being prescribed. How do outcomes differ based on whether someone's using oxygen? Dr. Berg at Genentech has shown associations with oxygen use and estimates of survival. And Dr. Joshua Mooney looked at differences between patients with similar GAP risk scores to find characteristics associated with people who might be listed for transplant compared to those who are eligible, but they're not listed. Very important issue with pulmonary fibrosis is that the diagnosis process can be long. Earlier and better diagnoses are critical to receiving the most appropriate care. So some studies have looked at the diagnosis process. What factors are associated with the level of confidence in diagnoses? Some regional differences were found by Dr. Sholin and authors. How often are multidisciplinary conferences used in diagnosing patients, and what factors are associated with this? This was examined by Dr. Bonnie Wang and others. Prediction models. Those are mathematical formulas developed to ascertain a person's overall risk of an event like a hospitalization or death over a certain period of time. And they can be very useful to clinicians to come up with a prognosis or to ascertain how aggressively to treat a patient's disease, balancing the risk of treatment that may have side effects or cause some kind of burden to the patient against the risk of not being treated. A variety of initiatives have begun for developing and improving current prediction models. The Prolific Consortium is looking at biomarkers and risk for potential clinical trial participants. The Precision Study is using biomarkers and whole genomic analysis to predict disease progression. Dr. Humphreys and O are looking at HRCTs combined with clinical data to make a prediction model. And Dr. King at Innova is looking at validating a prognostic scoring system that's already been developed using the registry participant data. So the pulmonary fibrosis maintains a list of published journal articles, posters, and abstracts. So I'm also at the end of my presentation, and I just wanted to direct everybody to where you can find more information about all the different types of research and links to papers and abstracts that have already been published. So just go to this website and scroll all the way to the bottom of the page where you'll see the words PFF Registry Studies. This is a screenshot of the bottom of the page here. So just look for these words, PFF Registry Studies. And if you look to the right where this other red circle is, that's where you can click through the different pages of the different publications that are out there. So I will finish my talk here. But before I end, I would like to give a big thank you to all the participants who give their time and have allowed us to use their clinical data, surveys, and biosamples so that we can improve life for all people with this disease. Also, thank you to site coordinators who collect and enter their data, and to the researchers who develop ideas and publish this valuable research. Thank you again for your attention.
Hi, everyone. I'm Ayodeji Adegunsoye from the University of Chicago. And today I'll be speaking on the topic, racial disparities in healthcare outcomes for those with pulmonary fibrosis. These are my disclosures. By now, we've heard a lot about the PFF Registry, and we're familiar with its wonderful capabilities as a resource for driving PF research worldwide, well, not just in the US alone. In the current iteration of the registry, the majority of patients enrolled are men, and almost half were over the age of 70 years at enrollment. Participants were predominantly white, as can be seen on the screen as well. Now, while this is probably the most robust prospective PF database in the country, we strive to improve every day. And our goal is to see more enrollment of females and minorities with numbers that approach that of the broader US population. So let's dive into some of these exciting observations from the registry. As we examine the racial distribution of pulmonary fibrosis within the registry, I'd like to first state that a lot of this work was performed with the help of Elizabeth Freiheit from the Saber Institute at Michigan, who provided wonderful statistical support in carrying this endeavor forward. Out of about 2,000 participants who self-identified based on the federally defined categories of race or ethnicity, over 3/4 were white, 105 were Black, 48 were Asian, and 124 identified as being Hispanic in ethnicity. For simplicity, we stratified these into the 4 main categories depicted on your screen. So just a couple of years ago, as enrollment in the PFF registry wrapped up, we made a striking observation. We found that Black patients were being diagnosed with pulmonary fibrosis and enrolled into the registry at a much younger age than those from other racial groups. We presented these findings at the ATS International Conference in 2019, but given that the registry then was still relatively young, we were limited in our ability to perform follow-up analysis of outcomes. But we went ahead and examined 3 large independent ILD registries to ensure that our findings weren't an artifact of who was getting enrolled in the registry at the time. So we focused on prospective registries that enrolled patients with diverse forms of pulmonary fibrosis in the Midwestern US, on the West Coast, and in the South, as well as an IPF-focused clinical trial dataset as well. Surprisingly, we found similar results. Black patients develop lung fibrosis almost a decade earlier than others. Perhaps the most intriguing question then is, does survival differ? Those of us who are familiar with epidemiological studies might expect that picking up a disease early could translate to longer survival times, regardless of therapy. And that's a concept often known as lead time bias. So we performed a multi-center study of over 1,600 ILD patients from Chicago and stratified survival by race. Interestingly, we found that African Americans had slightly increased life expectancy even after adjusting for disease severity. But perhaps the most striking finding was that although Black patients appeared to live for about 1.5 years longer than whites, their pulmonary fibrosis diagnosis occurred about a decade earlier, with a net loss of about 8 years, slightly more than 8 years. As the data elements in the PFF registry became much more robust over time, we examined this same survival phenomenon within the registry. Amazingly, our findings over the first 3 years were almost exactly identical to what we had found in Chicago. The longest transplant-free survival times were present in Blacks and Asian patients, and the shortest in white and Hispanic patients. In a composite outcome model that evaluated hospitalizations, lung transplants, and all-cause mortality, these differences between racial categories were much less profound. So we then elected to evaluate outcomes based on objective metrics that patients often ask. For the clinicians in the room, how many times have we had patients ask questions like, now that I have this diagnosis of pulmonary fibrosis, how long before I end up in the hospital? Or how long before I need to get a lung transplant? Or how much time do I really have left? So we assessed the age at each of these events for patients enrolled in the registry. And as you can see, a pretty consistent theme repeatedly was that Black patients with pulmonary fibrosis have the most unfavorable outcomes across board. Beyond age at diagnosis, Blacks were youngest at time of first hospitalization, at time of lung transplantation, and at time of death. As we think about potential reasons for differences in PF outcomes, a common question that often arises is, do genetic differences by race really matter? I think this really important figure encapsulates a critical issue, the lack of inclusion of racial and ethnic minorities in most genetic studies to date. As we know, racial differences in disease risk and mortality are important determinants of outcomes in population health. So, given these observations in Blacks, we undertook a PFF-supported study that assessed the effect of the MUC5B risk variant, that's the most common risk allele for pulmonary fibrosis so far, on the risk for developing the most severe form of fibrosis, the honeycomb fibrosis pattern that's often reported on CT scans. Amongst 364 patients who were evaluated with various forms of pulmonary fibrosis, the racial category with the least frequency of the MUC5B risk variant was found amongst Blacks. This risk variant was more common in older adults with PF, and most importantly, when present, it conferred a similar risk for honeycombing in patients of African ancestry compared to those of European ancestry. This might then suggest that the underlying genetics play a much bigger role than the racial category of patient assignment when we see these individuals in clinic. We then performed the sensitivity analysis of the PFF dataset using unsupervised cluster analysis to identify patient phenotypes based on how clinically similar they were at baseline evaluation. We found race to not be a critical factor for defining clinical PF phenotypes. Race wasn't as critical as we thought it would be. Instead, other factors such as lung function, that is forced vital capacity and diffusing capacity, sex, smoking status, diagnosis date or duration, and autoantibody positivity were all much more important variables. This again might imply that the effect of race itself may not necessarily be biological, but may be acting as a surrogate for other critical factors that put patients at increased risk. Now, during the course of this talk, I've suggested several times that socioeconomic disparities seem to be important in determining the exposure to predisposing factors for pulmonary fibrosis. An important factor here is air pollution. In the US overall, the air has become cleaner over the last 70 years, but these benefits are primarily seen in high-income areas with predominantly white populations. Unfortunately, Black patients and Black individuals tend to live next to transportation corridors and in regions that are close to toxic waste dumps and those factories that produce this waste as well. Occupational exposures are no different. Blacks are disproportionately represented in the service industry, in the labor industry, and we saw this recently with the heightened risk for COVID in Blacks and other minority populations. When we think of West Virginia and the Appalachian region, commonly called coal country, a lot of workers in this region are Blacks. But even beyond that, when we think about diesel exhaust exposure from truck driving, exposure to toxins like asbestos or coal dust or silica from sandblasting, cleaning services, and so on and so forth, many of these are particularly common amongst minorities and pose dangerously high risks for pulmonary fibrosis. So what can we do then, or rather, what should we do? We've come a long way in our ability to recognize and potentially mitigate several of these factors that lead to racial disparities in pulmonary fibrosis. These are factors that span the continuum from risk for pulmonary fibrosis and its diagnosis to the available treatment options, including lung transplantation and outcomes from pulmonary fibrosis as well. Identifying these risk factors is of prime importance, and perhaps more importantly is ameliorating them, which would result in an importance in changing this risk outcomes that we talked about. So this should be paramount in our approach. We should have more emphasis on looking at these issues from this perspective. promoting health equity, cleaner air, better lifestyles, access to quality healthcare, public education, and enlightenment. These are just a few of the interventions that will go a long way in helping to reduce these disparities and improve outcomes across board for all patients with pulmonary fibrosis. So in summary, pulmonary fibrosis and its poor outcomes remain common across all races, and the diagnosis occurs much earlier in Black patients who also have the most unfavorable outcomes. The increased risk for honeycombing from the MUC5B variant is similar across African and European ancestries, and we should focus on mitigating socioeconomic and structural inequities if we are to indeed improve lung health and reduce these racial disparities identified already. Thank you so much for your attention, and we'll take questions at this time. Thank you all again for joining us on day number 5 of what has really been an absolutely fabulous, fabulous summit. It's an exciting time to be in science and to be an active participant on what is currently a global movement to advance the care of pulmonary fibrosis. We've already gotten a substantial number of questions from our audience, and I'd like to begin with posing these to members of our stellar panel today. I'll start with one for Dr. Gibbons. So Dr. Gibbons, you very eloquently highlighted how important pulmonary fibrosis research is to the core mission of the NHLBI. And in your talk, you clearly articulated in the keynote session how concerned the NHLBI is about the detrimental effect of that delay in diagnosis. What do you view as the most pressing scientific advances that are needed to improve patient care for those living with pulmonary fibrosis today?
Oh, well, thank you for that. Well, I think they're manyfold. Certainly, earlier diagnosis is a key part of getting to the point where we can more effectively predict, hopefully prevent and or preempt or enhance the natural history and health of those affected by pulmonary fibrosis. So that's why we alluded to, for example, advances in new technologies, for example, in imaging. Similarly, as we get a greater sense of those who have a genetic predisposition to pulmonary fibrosis, getting a sense of their heritable risk through the technologies that exist now that can sequence someone's entire genome, gives us the hope that we may find certain genotypes that could be predictive of that predisposition. And so eventually biomarkers that may indicate either those at risk or those that may have a particular prognostic risk and course, those such that we can target our more aggressive therapies to those who have the most dire consequences and enhance both the maximal benefit by getting the right patient, the right intervention at the right time. Ultimately, that's part of precision medicine, and that's what we hope is part of the future for pulmonary fibrosis. Thanks.
Thank you so much. Yeah, there's been a tremendous amount of interest in biomarkers and genotypes, as you stated. So as a follow-up to that, what is the NHLBI doing to help support research in these high priority areas, if you may?
Well, certainly, as we alluded to earlier, our TopMed program, Transomics for Precision Medicine, is creating one of the largest resources in the world that combines genomic information with clinical phenotype information across the spectrum of heart, lung, blood, and sleep disorders, of which pulmonary fibrosis and other chronic lung conditions are a key component of the elements of TopMed. And that we're creating a cloud-based resource we call Biodata Catalyst, which really creates a sort of collaborative platform that brings all that data together, both the genetic information, the personal characteristics of patients, clinical characteristics of patients, increasingly mobile data that provides for patient-reported outcome data, environmental data to give a sense of the social and environmental factors that interact with genetics to predispose to conditions like pulmonary fibrosis. And by bringing together these multidimensional data sets at scale, we are very optimistic that we'll be able to further define the mediators of these diseases and really target another generation of therapeutics that can, again, interrupt or modify the pathobiology and hopefully arrest the progressive nature that is so common in pulmonary fibrosis.
Wonderful. Yeah, I and many other people are so excited, looking forward to the harmony that these different components are going to bring to bear on the care of our patients with pulmonary fibrosis. So thank you so much for that, sir. I'll switch to Elizabeth Freiheit. Dr. Freiheit, you spent the last several years leading efforts at SABR, the Statistical Institute that helps to navigate the enormous data that we have warehoused in the PFF Registry. And you've had a front row seat to witness the evolution of the data and statistical methodologies that have been used to analyze the data as well. So as the registry now moves to its second phase, how do you anticipate research will change with the new community and the care center registries that the PFF has been working on over the last few months?
Oh, thank you for that question. Yeah, it's very exciting what the new registries will allow us to do. For example, there's new questions that we cannot currently answer with the registry that we have now. For example, when the community registry launches, it's going to collect some really valuable exposure information, which we'll be able to combine with genetic information that will come from the care center registry for people who donate to the biorepository. And this could allow us to create combinations of component causes. In other words, causes that combine that lead to the development of some forms of pulmonary fibrosis. In other words, actually being able to figure out what causes pulmonary fibrosis in some cases. And if we can do that, that can lead to identifying ways to prevent pulmonary fibrosis, identifying people at greater risk of developing pulmonary fibrosis, early diagnosis, better ways to treat pulmonary fibrosis. So that's very exciting. Other new questions we can answer are questions about caregiver burden and the caregiver experience, family member experiences, and the new way we're organizing these two new registries, the community and the care center registry, is going to allow us to be much more flexible. We'll be able to bring on new initiatives to a population that's already consented and ready and waiting. They've consented to future research. The sites are in place. And we'll be able to, if people come up with cutting edge research ideas, it's not going to have to wait for an entirely new project to be implemented. We'll be able to roll out those types of initiatives. So I'm very much looking forward to the launch of those registries in 2022.
Yeah, that's very exciting. Looks like the registry is tackling the different facets of pulmonary fibrosis from the patient standpoint, the caregiver standpoint, the care provider, and the clinician standpoint as well. So I'll just ask a question that from the audience looks very similar to that. Along those lines, how many research projects are active right now? And when do you think more results will be made public?
So you saw from my presentation that those questions have been kind of building and building. We have about 40 separate research studies that are going on right now. And they've all gone through, most of them have gone through the phases of being submitted as abstracts and presentations at conferences. And many of them now are in the manuscript phase, which means they're being fully fleshed out, details, much more detailed information, much more inferences about the inferences that you can make from the research, and they're being prepared for publication. So there are quite a few of them. I imagine in 2022, we'll probably see quite a few more publications going. And when those publications are published in these leading journals, there will be a link to them on the PFF website, so people will be able to see what's been published.
I can't wait. Sounds like there's a lot in the pipeline waiting to be unleashed.
Yeah, I think there'll be quite a lot in 2022. There's so many that are very, very close to being submitted, and some are already submitted. So
yes, amazing. I'll pivot to Dr. Martinez, Dr. Fernando Martinez. So Dr. Martinez, you've had a really fascinating career over decades now studying this very disease, and you've contributed immensely to the wealth of knowledge that we have today in the fibrosis landscape. And I listened to your talk on precisions. I think that's just the latest chapter in in your illustrious career. So I'm going to ask you a question that came from the audience, and I'm sure a lot of patients on the webinar today don't really feel equipped to deal with a lot of issues that we're talking with. But a couple of similarly themed questions that were addressed to you include one on genetics. So what is the typical genotype for IPF? And is there any data that shows any effect on lung function in patients with cystic fibrosis who get inhaled NAC, that's N-acetylcysteine, as a mucolytic agent? So I'll stop there and just hear your response to that.
Well, Deji, thank you so much. And I'll pay you later for the laudations. That's not necessary. That's something only my mother would appreciate. And so, yes, I've been at this for 35 years and have been involved in a lot of these studies for a long time. And you know, Gibbons, I think that it's important for folks to realize how much the NHLBI has contributed over the course of the years to all of these endeavors. I mean, it's been decades that many of us have been involved with the lung division at the NHLBI in conducting all of these studies. And you're right, Deji, that the Precisions program, which is, I got to thank Gibbons there. He was a big proponent of that at the NHLBI for giving us an opportunity to sort of advance from what we've done before. And so the Precisions program, to me, has been such a fascinating component because there are so many innovative components to it, Deji. And I know Bill is quite familiar with this from the PFF, because not only is it an NHLBI endeavor, and I will get to these specific questions in a second, don't let me forget that, because I'm old and I'm getting frail now, getting the frailty phenotype here. And so there is an interaction between the NHLBI and the PFF, which includes all of the PFF sites and a subset of the PFF sites to conduct a clinical trial. So, Bill, that's an innovative approach to this component. It involves a foundation and a pharmaceutical company that are partnering with those groups to be able to conduct such a study. Also something innovative in this field for us. And I think most importantly, it is the entire program is proceeding with an approach that is the definition of a precision-based approach to therapy, in that we are taking one particular group of individuals with IPF that have a very specific genotype based on data from an IPF-NET study, Gibbons, a prior NHLBI endeavor for the IPF Clinical Research Network, that provides us an opportunity to test whether a very simple therapy that costs just fractions of $1 per month may actually have a benefit on key outcomes. That is like an oncology model of what is being done, and it is about as precision as you can get. So with regards to what we've demonstrated so far, one of the key questions that was asked was, what is the typical genotype? And that's more for you, Deji. This is more your area than mine. I'm just a simple clinical trialist. I'm not as smart as you guys who do a lot of the genetics. But my sense is there are multiple genes that have been associated with fibrosis. Some that are very well related to the risk of getting fibrosis, the mucin gene that David Schwartz identified, for example. And then the TALP gene, which you and your group, Chicago, aided in identifying. That latter genotype is what we're identifying. And what we're testing is whether this very simple therapy in 25%, that's the genotype that appears to be favorable for this particular therapy, whether this very targeted approach to treatment in a quarter of the patients with pulmonary fibrosis will exhibit a significant improvement in clinical outcomes is what we're testing. So we're testing this in about 25% or so of IPF patients for this one particular question. It's interesting because NAC, my other life is airway disease, COPD, where we've done large studies on NAC and COPD. There actually are studies in CF as well with regards to N-acetylcysteine, inhaled N-acetylcysteine, with suggestion of improvement in lung function. And very recently, in the last 6 or 7 months, Deji, I don't know if you were aware that there actually was a study of TALP, the same gene that we're testing in IPF, in CF, to see whether that actually potentially has the same kind of interaction. So for my CF colleagues, I encourage them to consider testing something very similar. So when you started your introduction, for some of us that have been involved in IPF research for the last 30 years, the field is completely different now than it was when I first got started. I mean, the approach to diagnosis, the approach to how we categorize patients and consider what to do therapeutically are drastically different than just 6 or 7 years ago. And my sense is over the course of the next 2 years or so, it will be radically different again in a good way. And I think that that's why we're all here and we're all sort of pushing ahead with this. It's really exciting times in this field. I mean, it's just dramatic how much better things are than they used to be.
Absolutely. That's really fascinating. I would say that the precision's approach is a game changer for us in how we meld genetics into the overall care for our patients. So thank you so much for heralding that effort. Another question that came in for you specifically was, so with your research and in partnership with the pharma companies, are we making significant inroads to hopefully finding a cure, finding drugs that either prevent or help to cure IPF specifically?
Yeah, so I think that the answer to that question is absolutely yes. Just sort of following up from my last conversation, in our prior major therapeutic approach with partnership with the NHLBI, the IPF network, we clearly identified what you should not do for an IPF patient because you can hurt them. That sort of changed the paradigm of how therapy was provided for IPF patients. And then in partnership with pharmaceutical companies, there are now 2 approved drugs that actually work. I mean, seriously, that is a dramatic benefit for us. There are now, I think I gave a lecture on this, Deji, recently, and I think there are 37 or 38 active clinical trials, including several phase 3 programs that are in the midst of being conducted in IPF. It's scores of therapies that are likely going to be effective. It's just dramatic what an advance this is going to be. And what I suspect will happen, Deji, is you and I will be dealing with combinations of agents targeting to the particular patient to ensure that a particular patient gets the best therapy for them. And the field of pulmonary fibrosis therapy, and it's just going to be in the next couple of years, is going to be even dramatically better than it is now, which is dramatically better than it was 7, 8 years ago.
Yeah, absolutely. Thank you so much for that. Thanks. That's wonderful insight. I'll take another question. Actually, this I think is directed to me. So the question goes, is there a comparison in these studies relating to racial ethnicity as to disease progression and survivability? Well, I'll start by saying that we live in a culture where race is often misconstrued as a biological representation of who we truly are. It's not. So just to make sure that people understand, race is more really of a complex sociobiological construct that reflects how an individual perceives their familiar origin, their cultural environment, and their genetic makeup as well. So when we state the term race, it's often a surrogate for these and other social factors as well. Now, importantly, we are finding that the effects of race may not necessarily be biological. We've thought about it in that context previously, but it really may be acting as a surrogate for other critical factors that put patients at increased risk. And it's important for us to tackle those factors because they also play a role in prognosis, in disease diagnosis, and in outcomes. So specifically for the question, yes, there's now data that shows that at least in the PFF registry and several other US-based registries, race and ethnicity does impact disease course. It does impact clinically important outcomes in pulmonary fibrosis. There's another question in the chat, and I will direct this towards Sean Craig. So Mr. Craig, talking about race, essentially, this has been your lived experience, right? All along the path from the diagnosis through treatments to the point of lung transplantation. As a person who's frequently categorized as other or mixed, what are your thoughts on patients of diverse diverse ancestries and diverse racial groups getting enrolled in the PFF registry going forward?
Thank you for that question. And yes, being of mixed race ancestry, it's one of those things that as I was first diagnosed with IPF back in 2015 through my lung transplantation, all of my doctors never really knew how to categorize me because of the fact that my biomarkers, et cetera, didn't really relate to anyone else. Here in Hawaii, where I live and born and raised, there's so many of us who are now dealing with ILDs or PF, and we're of such mixed ancestry that I tell people it's so important to get involved with genetic testing, get involved with the PFF registry. I first signed up the moment it opened. I signed up immediately because I knew that I was one of those others that didn't really have a category that I fit into. And I'm hoping that in the future, as more research is done through genetic testing with the PFF Registry, even with clinical trials, I tell all of my my constituents, please get involved, because the more we can get our self, recognized in all of this, the more we're able to find out the possibility of why things are happening to us, because again, we don't fall into any particular category.
Fantastic, very eloquently stated. I have a question for Dr. Martinez here again. So this is a question thinking about precision therapy and the approach to using NAC in these individuals. The question says, could taking NAC have a negative effect on an IPF patient? What are your thoughts about that?
Yeah, that is really, I'm glad you got to that question, Neji, because I think it's a crucial question to understand. If you remember the concept of this idea of precision therapy. So the idea is that you would take an individual patient, Sean, in your case, and decide that this therapy would be a good thing for you, but the alternate is there are potentially therapies that could be bad for you. And in fact, the precision therapeutic component, the NAC, inhaled NSC component, is based on the understanding that we believe, we'll see if we're correct, that NAC in a particular component of folks with that TOLIP gene is beneficial. But there is about a quarter of patients where our preliminary information suggests that it could be harmful. And so that's why just a drug as simple and as long-standing as N-acetylcysteine has to be tested in a very rigorous fashion to be absolutely sure that we know in whom it's good and in whom it's potentially bad. Both are equally important for our patients to understand.
Yeah, absolutely.
Precision therapy, Deji, that's the whole argument.
It's all about precision, moving from personalized therapy to more precision in our care of patients. Dr. Freiheit, this question goes for you. Is my data in the registry if I participated in the study in 2020, 2021? What are your thoughts about that?
You probably are not in the registry. The registry completed the enrollment in 2018. So you're probably not in the registry, but the good news is that in very early 2022, we're going to be opening up a new registry, first the community registry and then the care center registry. So, and that even that care center registry will not be completely closed to participants, because they will continuously try to enroll patients. So you will be able to participate soon.
Great, great. I'm glad our patients have the opportunity to do that. There's no closing the door to them. There's a question that is in the chat that states, what protein markers are being used now for IPF? I can take that. So I'll say you heard from Dr. Freiheit's talk about protein markers and the importance of proteomics in care for patients with pulmonary fibrosis. I'll say the world of proteomics is actually burgeoning now, and there have been several new identified protein biomarkers that have shown prognostic value, not just in IPF, but in several other forms of pulmonary fibrosis. Some of these include the matrix metalloproteinases, MMP7, chemokines like CXCL13, surfactant proteins, and other high-density lipoproteins. Now, while many of these are of tremendous research value, none of them have been guideline approved for the clinical diagnosis of IPF specifically. They have research value. They inform the diagnosis for us as we think about the disease and its course, but they really haven't been incorporated into guidelines for diagnosis purposes on a broader scale. So we're looking to see that coming on in future iterations of the guidelines as we get more and more data on those lines. A quick question as we round up here. We're approaching the end of our time, but I'll sneak in this question for Dr. Martinez as well. Thinking about precision therapy, a patient says, I have pulmonary fibrosis from connective tissue autoimmune diseases. Should my children be tested?
Yeah, that's a really great question. And the answer to that is we don't know. It is in part what the Pulmonary Fibrosis Foundation is now trying to identify for us in its next phases. As we expand beyond just idiopathic pulmonary fibrosis, it's very clear that there are individuals that have other disorders that can look very similar to what we used to call pulmonary fibrosis, that have significant negative ramifications for them from a quality of life perspective, maybe even from a survival perspective. And I think that the question that individual asks, which is such a clear, granular question to that individual and their family, is a question that I think we're all trying to tackle right now.
to be able to provide a good set of data that expands the concepts of how patients with fibrosis can progress, how we can identify those individuals and their families, and how we can hopefully target additional therapies. And as you know, Deji, we're planning a series of studies now that are hopefully going to provide a granular answer to that crucial question. I don't have a good answer for that yet.
Great, very, very clearly spoken and true to form. Thank you again, everyone, for another phenomenal session this morning. It's really been great to have you all and hear from you all as touching the next wave of what to expect in the field of pulmonary fibrosis. Hopefully for our audience, the excellent insights that have been provided from the panelists today has helped us improve our routine thinking and how we approach or navigate the research path towards a cure for pulmonary fibrosis. I'll pass this on to Bill Schmidt for the next session. Thank you all again and have a beautiful rest of your days.
Well, thank you again, Dr. Gibbons, and thank you to this wonderful panel. What a great session. We did have some technical issues that may have affected your viewing during this session, but please note that the talk will be made enduring and will be rebroadcast tomorrow morning. Tomorrow morning at 6 a.m. Central, to be precise, and the enduring begins on November 20th. Now I'd like to share some exciting news about PFF Summit 2023. We are very happy to announce our Summit 2023 co-chairs. Representing the healthcare provider community, Dr. Patricia Syme from Virginia Commonwealth University, Dr. Tracy Luckhart from the University of Alabama at Birmingham, and representing the patient community, Annie Lucas, PFF ambassador and lung transplant recipient from Charlottesville, Virginia. We look forward to working with them for our Summit 2023. We will present 4 outstanding sessions for our healthcare professionals starting at noon today, which will begin with part 1 of the Community Pulmonologist program and part 1 of the Nurse and Allied Health Network program. So please check the theater locations for each of these. There will also be sessions designed for patients and caregivers, so please check your programs for times. Thank you all very much.